welcome you all for this very interesting session on rare eye diseases. And we have the privilege of having among us very distinguished faculty and one of our dear friend from US, Ken. Ab nahi munda hai. Sorry, my Punjabi is not that great.
Don't worry, I'll let you off this time. Oh yeah, go ahead.
So you want to start the introduction? All right, please.
So welcome everyone. Can you put on the slides that are here? Okay, yeah. So we have a very esteemed set of panelists and guests today. We have Dr. Virinder Sangwan, who's chairing the session. He has over 25 years of experience in ophthalmic surgery and has also pioneered the technique of SLED for limbal stem cell deficiency. He has held multiple positions at the L.V. Prasad Eye Institute and has also been affiliated with the Harvard Medical School and Orbis International. He has multiple publications with a very significant H-index. We also have with us today Dr. Ken Nischal. He's the Medical Director of Digital Health and Chief of Pediatric Ophthalmology and Strabismus at the Children's Hospital of Pittsburgh, UPMC. He's also the Executive Vice Chair and Professor of Ophthalmology at the University of Pittsburgh School of Medicine. He pioneers in pediatric corneal transplants, and myself having learned a lot from him, with special focus on pediatric cataract, glaucoma, and anterior segment anomalies. He's also a member of the PGCFA Scientific Advisory Board and the co-director of the World Society of Pediatric Ophthalmology and Strabismus. Do we move on or we introduce? Okay. We also have Dr. Shirisha Senthil. Welcome, ma'am. She has over 22 years of experience in glaucoma, specializing in refractory adult and pediatric cases, and glaucoma drainage implants and also secondary glaucoma. She heads the glaucoma service at the L.V. Prasad Eye Institute in Hyderabad. With multiple publications and chapters to her credit, she has also won significant awards and recognitions. Welcome, Professor Neil Lagali. He is a researcher in corneal disease and regenerative medicine and biomaterials at the Linköping University of Sweden. He leads a group focused on keratoconus, diabetes, AMD, aniridia, dry eye, corneal infections, and also post-COVID eye problems. His research focus uses surgically induced and transgenic models for conditions like keratoconus and corneal neovascularization, and is also involved in multiple European projects on corneal diseases. With multiple publications, he is also the associate editor of the Ocular Surface Journal. We have with us today, welcome, Dr. Rajni Bhattu, ma'am. She's the medical director of the Center for Eye Genetics and Research in Bangalore. She's a PhD in inherited retinal diseases from Maastricht University in Netherlands and has received the gold medals during her MBBS and AFER Arvo Visiting Researcher Award in 2009. She specializes in genetic eye diseases and has also completed an ICO fellowship at Moorfields Eye Hospital, London. She has founded the Center for Eye Genetics and Research and also co-founded iStem, a leading retinal stem cell research company in India. And we have Ria. She is a certified level 2 genetic counselor with subspecialty experience in ocular genetics. She has a master's in human molecular genetics from the Imperial College of London and is trained in genetic counseling. She's associated with us for quite some time, and she leads the genetic testing collaborations with Indian and international laboratories and patient care. She's also pursuing a PhD with us in affiliation to the Manipal Academy of Higher Education. That's me. I'm a pediatric cornea specialist by training, and I've done a long-term cornea fellowship, and now I'm very actively working on the stem cell projects and the research that is happening in cornea. Dr. Shelja, she's a senior surgeon in the pediatric ophthalmology and strabismus and neuro-ophthalmology at SCH. She leads our ocular genetic services. She has a very good experience and a very significant experience in pediatric cataract surgeries and oversees the pediatric services at our peripheral centers as well. She has multiple awards to her credit, which is the Arvo Developing Country Eye Researcher Award, and is the project director for a USAID grant focused on child screening. Also the awardee of an Indo-US grant for studying match genetics, and has multiple publications to her credit. Welcome. With this, we would invite Dr. Ken to please start his.
Thank you. Thank you very much for asking me to speak in this. If we could start the timer so I know where I am. And if I could have my...
Can we change the slides, please? Can we have Dr. Ken Nischal's slide and switch off this presentation?
Okay, we got it. Okay, so it's not an easy subject to cover in 10 minutes, okay? Innovative solutions for rare eye diseases. So what I've decided to do, if this will go forward, let's have a look. Guys, it's not, okay, good. These are my financial disclosures. None of them are really relevant to the talk. So the first thing I would ask you is, be curious, not judgmental. If you see a child or a family with different things that you've never seen before, don't say, oh, it's too difficult, or I'm not sure what's going on. Be curious as to why you are seeing what you're seeing if you want to try and make the diagnosis or understand a rare disease. Innovation at its core involves challenging the status quo, thinking outside the box, and taking calculated risks to drive progress and achieve breakthrough outcomes. So I want to show you some examples of how I've been lucky enough to be involved with some of those things. I'm going to mention very briefly that Hannah Skanger, who is my genetic counselor, and I, together with several other people, did a genetic educator training course. We trained all these centers, their optometrists or technicians, to take a 3-generation pedigree, and we did that in 2014. 11 years ago, we introduced the ability to take pedigrees when it was not being done in any of these centers. And I know for a fact that part of why RIA, yes?
Just make more comments. And this impact of that training, Ken, you are seeing now. You are seeing cell jars. You are seeing GORA and LVP.
And we're seeing RIA.
RIA and yes. So you're seeing that impact coming and changing the patient care.
Thank you. That means a lot. Thank you. Thank you very much. But so I'm not going to talk a lot about that. It was a very detailed. We have, if you are interested in your area to do something similar, if you go to the WSPOS website, we have that ability to repeat that, perhaps a little bit better than we did it the first time. But thank you so much for that comment. Okay, here are the examples. If you want to look at rare diseases, look at a rare disease and then see if there's a generalizable application. Let me give you an example. This is a paper we wrote when we were looking at children with certain mitochondrial diseases. We noticed, and again, this is being curious, that when you looked at the cornea, something was not right. They had changes on the cornea that we were used to seeing in Fuchs endothelial dystrophy. See that on the OCT? And the question was, why is this happening? And this is a specular microscopy. This child is only 9 years old, and look at the cell count, look at the changes that you see, that you would see in Fuchs. Well, it turns out that if you look in the literature, there is reports of mutagenesis of mitochondrial DNA in Fuchs endothelial corneal dystrophy. And when you look at that DNA change, it's the DNA change that occurs most commonly when you see Kern-Serre syndrome or Pearson syndrome. So in the cells of the cornea, in adults with Fuchs, they have the changes that we saw in children with Kern-Serre syndrome. And here's another one which shows that there are degenerated mitochondria even when you have Fuchs in a COL8A2 mutation families. So based on that, and based on the work that we did, there are several studies going on now to see if you can rescue adult Fuchs with either coenzyme Q or preparations that are topical but are like coenzyme Q. So again, look at a rare disease that you can apply generally and you'll get more funding. What about large networks? Again, I am privileged to have been part of developing the Bodia Eye Consortium. Now, the Bodia Eye Consortium is a consortium of eye hospitals. They're all listed there, and you can see where they are up on that map. What is interesting is this. By having similar proformas that you fill in when the patients come in, you can then publish a study where the dropout rate, even though it's retrospective, is very low because everybody's filling the same proformas. But let me explain one other thing that's really important. We published this paper, and there was a very important point. It turns out that there is a difference genetically and anthropologically between ancestral North Indians and ancestral South Indians. Ancestral North Indians genetically and anthropologically come from 4 regions of the world: the Caucasus, i.e. Eastern Europe, the Mediterranean, North Africa, and the Middle East. So that means that when you do a study in North India, the genetic makeup is generalizable to the whole world except for sub-Saharan Africa. So if you are going to do studies, it is the only region in the world where that is applicable. So now, if you want to do a rare disease study, you do it in North India, you do it with a consortium, and this is the first paper we published. Shelja is in the audience, Suma Ganesh is in the audience, and this was clinical spectrum of non-syndromic microphthalmos, anophthalmos, and coloboma in pediatric population. We got 532 cases in just 12 months. The only other study to have numbers close to that, 312, took 16 years from North America. So think about the power that you have by forming a consortium of eye hospitals that work together. And Umang Mathur is really, he made sure it happened, but we had lots of morning meetings, if everybody remembers, 6 o'clock in the morning for me, a little bit later for you guys, where we started the Bodhia Eye Consortium. So think about large networks, because you get large networks, and as Shelja has proven, you can get funding that is internationally recognized. What about developing large disease-focused studies? This is harder, but again, I want to show you this. LCHAD is a very rare long-chain fatty acid disorder, and they get changes in their eyes. So a whole group of us right across America and in fact Europe got together and we collected all our cases. And what we showed for the first time was we showed firstly the natural history. If you look at A, this is how the patient presented in the teens, and by the time they're 30, look at the atrophy that they get of the retina. And you can see in cross-section, you can do OCA, you can do photoreceptor en face. You don't need to do that. The images show very clearly that there's a change. But this is the power of a natural history study. When we looked at the number of children who got, or the number of patients who got that horrible atrophy and lost vision, if there was early diagnosis, you could prevent it. And because of this paper, they started newborn screening in Europe for this condition, which never existed before. Okay? So again, large disease-focused studies. Collaborate with non-ophthalmologists. This is the most powerful thing that I have done ever. The more I work with non-ophthalmologists, the more I understand eye diseases. So, this is the first paper I published with Nathan Clark. Subterranean mammals show convergent regression in ocular genes and enhancers along with adaptation to tunneling. Let me explain what that means. All of these animals, over the last several thousand years, have lived underground. If you live underground, which organ do you not need? Your eyes. So what we looked at, and this was Nathan, and I met Nathan in a coffee shop, right? I met him in a coffee shop. He works at the university, and we started talking. So if you look at, let me show you a little bit better here. If you look, ROM1 is GNAD1 is a retina-specific gene. And when you look at it, everything in red means that there is a much higher rate of mutation. So even evolutionary biologists believe that if you don't need an organ, the mutation rate, the daily mutation rate goes sky high because the body's trying to get rid of that organ. And through a series of experiments, we showed that in subterranean animals, the visual perception genes are have much higher rate of mutation. Why is that important? Well, it becomes important because we now have identified a whole bunch of genes that some of which we know are to do with the eye, and some we don't know have anything to do with the eye. But we know they're mutated at a very high rate. So when we see a rare disease and we don't have a reason genetically, we start to look at the genes that are being mutated at a high rate in the subterranean mammals that might be also present in the patients that we're seeing with no diagnosis. In this way, we are then able, through convergence and looking at enhancers, identify genes that become putative genes for eye conditions. And in fact, we just published our first paper where we found a gene for congenital aphakia that was not due to FOXE3. So it's really important that you think about working with other people other than ophthalmologists, because that way we can actually make great progress. And on that note, please join the International Congenital Corneopathy Network, and there'll be a virtual meeting soon. That's the QR code. Register, and let's see if we can try and cure some more rare diseases. Thank you.
Thank you very much, Ken, for a very fascinating talk. Thank you very much. Please come here. And thank you very much for mentoring. And this both the consortium is a brainchild mentored by him, and Cells and Ria and Gora. It's all your students. They are doing fabulously well. Thank you.
Thank you.
Next, we'd like to invite Neil, our friend from Sweden, and he will be talking about limbal stem cell deficiency and rare ocular surface disorders.
Thank you so much. And I'd really like to thank Professor Sangwan and Ken for inviting me to speak today. It's really a pleasure. So we can just start the second talk.
Can we have the slides for Dr. Legali?
Great, thank you. So I'm going to continue this session talking about research and therapeutics for limbal stem cell deficiency, especially with focus on rare ocular surface disorders. So continue the session, but talking a little bit now, going back to models for understanding the disease. But we think of congenital aniridia as a model disease for limbal stem cell deficiency. And as we may know, congenital aniridia is a rare inherited eye disease. It can also occur sporadically, but it's a whole eye developmental disease. And it stems mainly from mutations in the PAX6 gene that control ocular development in utero. And there have been to date more than 500 different unique mutations in this one gene that have been linked to the aniridia phenotype. So it's a highly conserved gene. All the way from fruit flies up to humans. And the mutation is causing developmental delay or abnormalities in most of the eye structures, so including iris, macula, trabecular meshwork, lens, muscle, the extraocular muscles, as well as the cornea. Our interest is for the research is focused more on the cornea. And we developed this grading scale, clinical grading scale for aniridia-associated keratopathy. And what you'll see is there is a progressive invasion of conjunctiva to cover the cornea. So it's a pannus that's growing slowly over the cornea, and then in the end stage it results in a thick fibrovascular pannus. So to understand this better, we have to go back to the lab and look at models of limbal stem cell deficiency. And right now in our lab, we have both traumatic and genetic mouse models. For the traumatic model, it's a total epithelial removal using alcohol and the alder brush. And we also have genetic models as well. We've just established the P63 mutant mouse model, and that's crossbred with a PAX6 Cree promoter, so that the P63 mutation is expressed only in the cornea. And that is a model of the AEC rare disease. So that's ectodermal dysplasia and cleft lip syndrome, lift and palate syndrome, where they also have a progressive limbal stem cell deficiency. And in addition to that, we have the small eye PAX6 heterozygous mouse model, again with progressive limbal stem cell deficiency. So looking at that model, which is a model of congenital aniridia, first developed by Elizabeth Simpson in Canada, and my PhD student Dina has worked extensively with this model and characterized this model. And it's interesting because it allows us to look at it's a very good model for the human progression of limbal stem cell deficiency, which takes decades in aniridia patients. So it occurs here in the model relatively slowly in a mouse model over several months. And you can see that here compared to the wild type in the top row. The middle row shows a progressive conjunctivalization, and then the bottom row is more of a delayed development. And this is important because it gives us a tool to look at what's happening over time, how this limbal niche is changing over time, and also a potential way to test potential therapeutics. So there's a window of therapy. So looking at this model, we see that there's a lot of different pathologies in this model that go hand in hand with the limbal stem cell deficiency. So we have a corneal thickness is pathologically low. And if you look at the histology images there, you can see that the epithelial thickness is also significantly reduced. But not only that, the basal epithelial cells have completely lost their columnar structure, and they're no longer producing epithelial basement membrane, so there's no collagen 4 production. And then you can see there in the fluorescein staining that because of this, the epithelial barrier function has been lost. We're looking at also stem cell markers in this model and comparing to controls, and we see that, of course, the PAX6 expression is less because of the mutation, but also the P63 expression is reduced, as well as GPHA2, also a marker, stem cell marker. And this goes hand in hand with a rise of the conjunctivalization markers like MUC5AC. But interestingly, none of these markers are really disappearing. So we believe that even in advanced aniridia, the limbal stem cells are still present and probably functional, but they're just being overwhelmed by the chronic inflammation, chronic wound healing response. To look at this even in a deeper way, we're doing single cell RNA sequencing studies now with these mice in the mice corneas. So each dot here on these UMAPs represents a single cell in the cornea, and you can see, and these are grouped and color coded based on transcriptomic or gene expression. I've highlighted here in circles the brown cells. Those are the limbal stem cells. And you can see that in the wild type, there are kind of two populations of these. One population is sort of really exclusively expressing the stem cell markers, whereas the other population, they're overlapping with the orange cells, are more like the orange cells are transient amplifying cells. So they're looking a little bit more like transient amplifying cells. And then when we go to the right, here, we see that in the aniridia case, those stem cells are now overlapping with stromal cells. So that could be, there's a transformation of those into more stromal-like, as well as the orange cells on the right. Those are transient amplifying cells, but they're also changing. They're expressing more keratinization markers. So the way we do this, and this is work that has been done by my postdoc as well as a collaborator, and we look at these expression profiles in the different cell populations, but we look at a whole panel. You can see at the bottom there, it's a whole panel, like 20 to 30 genes. So we know that a cell is not defined by just its marker profile for one or two different markers, but it's really a whole range of markers. And so this is how we're starting to think about cells now and different cell populations in this way. It's a very complex phenomenon. And the next sort of where we're moving towards is spatial transcriptomics to give us also the spatial information as well about the expression of different cells. And so we've been doing this with both wild type and the PAX6 heterozygous mice. And if you think about this, this is like we take the cornea. Right now we look at the cornea, and if you see this histology picture, we can get the exact same section. We can get information on the transcriptomic expression of every cell in that section. And so it's sort of color-coded here, and we can look at it as in terms of clusters of cells that are expressing the same similar transcriptomic profiles. And you can see there that if you look at the epithelial cells in the cornea, it's quite a heterogeneous population in the epithelium. They're not all like basal epithelial cells or one epithelial cell type, but they are expressing very different different genes. And so this is just a blow up to show you in each dot here. It's a very high resolution. It's each dot is representing we can get the transcriptomic information from that point. So it's a transcriptome at 2 micron resolution, and this is going to allow us to study cell heterogeneity and also stem cell heterogeneity. And we're doing this in the wild type and the PAX6 mutant. So we're really going to try to uncover what is the role of this transcription factor, PAX6, when it's mutated. Now just quickly jumping over to therapeutic studies, we're looking at possible therapies for limbostem cell deficiency, and this was an example of a gene therapy with an antisense oligonucleotide eye drop that was given to the aniridia mice for 90 days, and then it was stopped. So this is just giving the vehicle, so this is the control. We can see the progression of the keratopathy here. Giving the drug, we still see vessels, but it's quite a bit suppressed by the drug. And when we quantify this, we can see this suppression, but it works only in an early stage of the disease. If you stop the drug, the progression starts again. So we're looking also into this in the mechanism of action using transcriptomics. And we found that doing the bioinformatics analysis that this works through suppression of MHC1 and 2, as well as CD8086, so those are the dendritic cells, and also macrophages. And interestingly, the transcriptomic analysis gives us insights into new genes. So for example, we found this gene LARS2 that's highly upregulated. And when we looked at this is a gene that's involved in the mitochondrial protein translation, enhancing oxidative phosphorylation in the mitochondria, then upregulating ATP in the epithelial cells, which then promotes epithelial wound healing. Just to conclude, we're looking also now at the power of stem cells in terms of bone marrow-derived mesenchymal stem cells, but they're the extracellular vesicles that they secrete. And we know that these have immunomodulatory and wound healing properties. And we're looking at subconjunctival injection of these in LSCD models. But we're also priming those cells, so we're cytokine licensing or priming them so that they'll have more of a immunomodulatory and wound healing effect. And now also again looking at the multi-omics, we're characterizing these EVs, and what we see is, first of all, from different donors, you can get a different profile of microRNAs and mRNAs, so it's really donor dependent. And also shown there in the heat map what happens when you license these cells. So licensing them, cytokine licensing them, is changing their transcriptomic profile. And I'll just conclude with saying that, yeah, we're doing the multi-omics to look at microRNA to mRNA interactions to see what's the most important for these EVs. And with that, I would like to thank you for your attention and thank our funders. Thank you.
If we have one question, we have time for one question. Do you want to come up to the microphone? Just say your name and who you are.
So I am Shelja. So I always wondered, I've asked this to Dr. Sangun also before, that while the other limbal stem cell deficiencies are treatable fairly, remarkably with SLET and CLET and other procedures, why is it that aniridia is so resistant?
Well, I mean, it's a bilateral disease, first of all, so we don't have any healthy limbal stem cells, but it's also a disease of chronic inflammation in the cornea. So they have this hypothesis, right? The seed and the ground. So you could plant the seed, the stem cells, but if the ground is not fertile and the ground is not right, then it will never survive. And that's what we see in aniridia. So we see this enhanced immune activity that makes it very difficult for transplanted cells to survive.
Yeah, I would just say I'm so glad that Neil has done the basic science, because for many, many years now, as soon as I have an aniridic child, even a baby, I'm very aggressive about using Restasis. They have to be over 1 for me to use Restasis, FML, and lubrication, including punctal plugs. And it really slows the progression of the keratopathy. So my first one I started in 2001. He was 4 years old at the time. And I'm so pleased to say, actually, he's in London, and my colleagues there are going to publish him as a case report. He's got no keratopathy. Even the pannus that you see has regressed from the first picture. So I've asked them to publish it because I'm not in London anymore. But I'm so
Yeah.
glad you published the
Early.
work, because that had been
Early.
my feeling, that the normal environment was being destroyed. The
Yeah.
palisades were being damaged.
Yeah, and I think we see even in the patients, but in the models as well, we see the earlier the better.
Yeah.
Yeah. this
Good,
disease.
great talk.
Yeah,
Thank you.
Can I ask a question? So we also see that clinically some aniridia do develop keratopathy and some of them don't. Some of them don't because we have children who are now adults and they are okay. So is there anything different in the genetics of these patients? PAX6 itself can have heterogeneity?
It depends on the mutation. We haven't figured out, I don't know if your group has, which mutations lend you to the keratopathy yet. But with the work Neil's doing and other people are doing with the rare organizations in Europe, I think we'll get that answer in the next 5 to 10 years, in my opinion.
Yeah, we published that it's highly dependent on the mutation type, so that you will get, like, for example, missense mutations are giving a less aggressive phenotype, and also the non-PAX6 cases. So that if you're not coding, if it's not in a region that's coding for the protein, then it's going to be a very mild case, for example. And then the whole chromosomal mutations and deletions, those give the most aggressive phenotype. But we're not down to the individual mutation level yet. We don't know.
Fantastic. Thank you so much for an amazing talk. So it gives me great pleasure to introduce Dr. Sirisha Senthil, who's going to talk about rare presentations, syndromic associations, and genetic implications of pediatric glaucoma.
Thank you for the invitation, Dr. Sangwan, Ken, and Neil here. So I'm going to speak to you from a clinician's perspective. So typically, what is it that we are looking at? What are the phenotypes like? What, some of these diseases that are associated with syndromes, what is the role of genetics and challenges in the management? So this, I would say, is a landmark paper that we published from India. 1,743 eyes of 1,155 children of new childhood glaucoma cases in a year. This is what, like what Ken was saying, would take a few years in the rest of the world put together to get. So we have a huge volume of patients, and we actually need help to be able to treat them, identify the conditions, and treat them better. So of this, close to 20% have ocular anomalies, and this is an underestimation. Close to about 4% have syndromic associations. Remember that syndromic associations is an ongoing process, right? As a baby, they may not even have a manifestation, and as they grow older, you will be able to pick them up maybe when they are in their teenagers or even older. So this could grossly be an underestimation as well. So when you look at pediatric glaucomas, there are phenotypic similarities and there are huge challenges with diagnosis. All these are congenital glaucoma, enlarged corneal diameters, elevated intraocular pressures. Some of them, you may see a HAPS trias, some of them have some kind of a keratopathy. Ultimately, we treat them with a similar kind of surgical intervention. Some clear very well. One surgery is enough to make things better for the child. Some of them don't clear. Pressures may come down, but the cornea never clears. May need a keratoplasty. Some of them may clear with a keratopathy in the middle. Some of them, you may be able to pick up a diagnosis only after you treat and the cornea clears. And this was this mother who also had actually a familial aniridia. And this is the cornea after it cleared. If you see a peripheral prominent Schwab's line, and then you go back and see there's some kind of change, like a bite-out spot-like picture that you see. And now you know that these eyes are phenotypically, to begin with, maybe similar. As they progress and as you treat them, they may end up having a different kind of a picture with time. All of them have different kind of genetic mutations. But if I just showed you that first picture, panel, with all hazy corneas, they're all not primary congenital glaucoma. Remember that they can have similarities, but only with time we will understand. Now, as a clinician who was not into genetics, we never had the opportunity a few years ago. I would say that yes, this eye clears well, and this eye unlikely to clear, may need more surgeries. As a clinician, I've learned to understand that. But now you can actually put that genetics together and say, when you see these eyes, now you can actually tell that this is this type of a mutation, and this is likely to require more number of surgeries that can only be proved with a genetic testing. Syndromic and systemic associations, like I said, one of the commonest syndromes that we see is Sturge-Weber's, and this is not difficult for us to diagnose, right? It is an encephalo-trigeminal angiomatosis, and usually it is unilateral occurrence, and ocular involvement is quite significant. There is its sister or brother, you may call, is a condition called phacomatosis pigmentovascularis, where you have Sturge-Weber's-like picture with also the pigmentary nevi that are present. And you see that this is the kind of appearance. You have a bilateral presentation often, you have pigmentation, and this is a multi-system condition where it's not just the encephalotrigeminal involvement, but you can have the rest of the body being involved, the trunk as well as the limbs. Why is it important to distinguish these two conditions? It's important, very important, because we know that this is a bilateral disease. At least Sturge-Weber's, though it is difficult to treat, you have the other normal eye. And this condition, you have bilateral involvement. They have significant more systemic involvement. Recently, we published this article where they have significant anemia because of the extent of hemangioma that they have. In fact, we have children with 4 or 5 hemoglobin levels who may require multiple interventions before you even go and touch the child. Systemically, they need to be stabilized. We have children who have multiple episodes of hemiparesis. So, the child doesn't come for the next 4 months, and then I ask the mother, what happened? Why were you delayed? No, he was admitted in the hospital. He had an attack of hemiparesis. That is at the age of 3 and 4 years. So, they have significant higher episodes of hemiparesis, and surgically, these two diseases behave very differently. Sturge-Weber's, they do much better compared to phacomatosis, and also the number of complications that they have post-operatively are also higher. So this is again unilateral congenital glaucoma. We know that 80% of primary congenital glaucomas are bilateral. You may have a unilateral presentation, but when you have a presentation like this, always think that is something beyond just the PCG. So you can see that little bit of thickening on the lid and little bit of pigmentation you see on the temporal side. Examine the face as a whole. Typically, they have pigmentation on one half of the face. This is NFN-related neurofibromatosis. It's important because they can have systemic involvement, systemic malignancies. And also remember that this disease can progress relentlessly, and we may lose the eye as well. But what is also very important is recognizing the condition and diagnosing it is important, because for more of these monogenic disorders, it's possible that there could be some treatment option available. And this is a very exciting thing that we found, that selumetinib is a drug that is possibly used in very severe type of plexiform neurofibroma which are inoperable, possibly try to decrease the proliferation of the tissue, and this can also be used in congenital glaucoma. So this was a premature child that was referred, a premature baby with congenital glaucoma, came all the way from Mumbai, had the surgery done, cornea cleared very well. We were very happy. We thought it was a premature child along with glaucoma, that it's taking time for the child to recover. By about 6 months, the child would not have an eye contact despite his disc not being very bad. And with time, the disc cupping kept progressing despite the pressures being under control. And then later we came to know that this child was in fact asked for a neuroimaging and had a genetic testing done. Born to non-consanguineous parents, homozygous TREX1 mutation, which coded for Aicardi-Gutierrez syndrome. And this is a very, very difficult disease to treat. And now what next? So what is important? In fact, I learned this from these parents, that they went through an IVF. Parents chose to undergo pre-implantation genetic testing and screening and had an implantation of an embryo that was normal, and these parents have a normal child now. So it's very important not just to diagnose, to be able to help the child with the diagnosis, but also help the parents with their future generations as well. So this is again a very interesting and a very a topic that I like a lot, about ectopia lentis. All ectopia lentis, whether you like it or not, are associated with some systemic problems. So if you have a 12-year-old who comes with high myopia, ectopia lentis, all of them are not morphins. They can have several other conditions that are associated. So we need to remember that sometimes clinically we may be able to recognize it, but most often we may not be able to. And remember that some diseases can have serious ocular as well as systemic problems. So it's very important to be able to pick up all these changes, to be able to diagnose the conditions appropriately. Marfan's, Weil-Marchesani, homocystinemia, this is again not very common, but yes, only if we look for them, we will be able to identify. Just fasting and waiting for a GA, you may lose the child for a deep vein thrombosis. So we should be very, very careful in what we diagnose. Other condition with ectopia lentis, referred for a congenital glaucoma. Don't do glaucoma surgery in them, you lose the eye. It is an LTBP2 mutation with a subluxation or a dislocation of the lens. All they need is a lensectomy. Trabolsis syndrome, this is again something that we don't understand much about, but we know that these eyes progressively, the sclera thins and they develop spontaneous blebs, hypotonia, and you lose the eye. And if you are not careful, just with your surgical intervention, you may be worsening the condition. So what is important is systemic evaluation is very, very important. And we, like I said, we are a learning. With time, we will be able to pick up those conditions better. But if there is any situation where you're treating it with your best ability, but if it is not responding to treatment, or there are features which are slightly different, ask a question, go back, re-look at it, deep phenotype, or maybe get help of the genetic investigations to be able to identify the condition better, then we should be able to help them with their systemic evaluation and, like I said, help the family with the child as well as with the future generations. So to summarize, it's very important to recognize the conditions and to be able to take help of genetic testing and parental and sibling screening as well to be able to diagnose, appropriately treat, as well as follow them up. Thank you so much, and all of you are welcome for the IPGS 2025 in December 12th and 13th at Madurai.
Thank you so much for a wonderful talk. I think we have time for a question. Any questions?
Can I make a comment?
Yes, of course.
So, thank you, Dr. Sirisha. It was really an eye-opener for me. No, this is just a comment on the pre-implantation genetic diagnosis. So, I'm a retina specialist. And so this is now quite commonly done across the country in, many, at least in all tier 1 cities. Especially when the parents both are carrying a mutation, or they have one child and they're worried about the second child carrying the same mutation. So an IVF is done, and then so a laser guided biopsy at an 8-cell stage. It's a trophoblast stage. So at that stage, a laser guided biopsy is done, and a mutation analysis is done of those. So then they decide, which embryos can be put in. So I have at least a few patients, parents who've undergone this and have a successful child with no mutation.
Yeah, it is amazing. And for me, this was the first patient that I came to know about. And then, so what is very disheartening is that when one child, at least in our part of the country, there's one child with congenital glaucoma blind, and then the mother doesn't show up for the next 2 years. And then they come with 3 children, all 3 of them affected with the same condition. And unfortunately, all of them in the lower socioeconomic strata. So one is we need to, first of all, treat this child, and we should be able to tell, because they think that, this child is blind. We need to have one more baby so that they can take care of the other child. And not realizing that the same problem is happening in the next generations as well. But this was an eye-opener to me, and now I tell this. I take time to tell the parents that if you are planning the next child, you're telling us, and we will be able to help you. Yeah.
Thank you. I think for the sake of time, we have to move on. I'd like to introduce Dr. Rajni Bhattu, who's going to talk about update on research in the treatment of retinal degenerative diseases. Yeah, we're good.
Thank you so much. Yes. Oops. I think it's a topic on retina. We've covered cornea, glaucoma, pediatric ophthalmology, and
Just give us 1 second. Can we have it up on the screen?
Yes, we do. Yeah, we got it. Yeah. So, so there has been a paradigm shift in treatment of IRDs, at least the research into it. Well, I can't say that we have the treatment because some of these are not in India yet. So 2017, Luxturna for an RP65 mutation got approved. Unfortunately, Novartis had planned to do it, launched in India, and they pulled out of it last year. So we now have phase 1 trials of gene therapy for other, Leber amaurosis, and the latest in the block is the nanoscope optogenetics. That seems to be very promising. So when gene therapy, we talk about mutation-specific conditions, whereas when you talk about the cell therapy or optogenetics or Neuralink of Musk's Neuralink, these are all going to be mutation-independent approaches. So I'm just going to talk about the cell therapy and the landscape as we have today. And so a lot of the initial work was being done on using embryonic stem cells. These are obtained from the blastocyst. And so when Professor Shinya Yamanaka from Japan showed that you could take a peripheral blood sample and convert them into stem cells. They're called induced pluripotent stem cells. This sort of changed the world of stem cell research. And so most companies or academics that are doing research today mostly are working on AMD, because we work on the retinal pigment epithelial cell as a first step before we go into photoreceptor diseases. So that's the outline. The average timeline that it takes for a proof of concept to go into the commercialization is between 15 and 20 years. So it's a lot of hard work, a lot of money, and a lot of effort, of course, that we have to sustain to get to that point. I'm going to spend the next few slides quickly on the journey of iStem. iStem is a startup based out of Bangalore. We are a stem cell-based company specifically for retinal diseases. We again chose to treat the first disease as geographic atrophy or dry AMD. It's an iPSC-derived RP suspension. So a lot of work happens in the background before we can even say that this is available for us. So what we do is, this is a 120-day protocol before the iPSC becomes a bunch of, RP cells, pigmented cells, in a dish. So once that's done, you have to have multiple animal studies. Most of our small animal studies were all done at OHSU in Portland, and we chose the RCS rat. The implantation here is a blind procedure. It's a transscleral delivery, unlike what we do in a human eye. And we had fairly good functional results and structural recovery. And so, sorry, I'm just going to skip this for the sake of time. So we've done, that's a summary of all the animal studies that we've done since 2017 to now. And once we had all of these, we have to show that, the cells are safe, there are no tumors, toxicology studies. So incidentally, the toxicology studies were done at Dabur Research Foundation, here. And the next big problem to solve is you have all of these cells, you know that they are safe and they're efficacious in the animals. How do we deliver it in the human eye? So we collaborate with Singapore Eye Research Institute, to understand how these can be done. We sort of did multiple experiments to understand. So while surgically it is a subretinal delivery, there are problems associated with it because during the process of delivery, the cells that reflux can create an epiretinal membrane, and that is one of the biggest problems that this field is facing. So, so that's the regulatory roadmap. A lot of work that goes in, including your CMC, your GMP manufacturing, a lot of work before we say that we're now ready to submit. So we submitted to the DCGI or the interregulator as for approval for IND, an investigation new drug approval for a phase 1 study. And we got an approval 1st of April last year. And so we started our first in human studies. These are phase 1 studies. Phase 1 studies are mostly for safety studies. We've got to show that these are safe in the human eye. So we had a dose escalation study of, it's 9 patients across 3 plus 3, 100, 200, and 300,000 cells. And then 3 participating sites, L.V. Prasad in Hyderabad, All India Institute, RP Center, and in Delhi, and Sri Ganapati Retralaya in Jalna. So all of these are immune suppressed patients because we did for 3 months because these are allogenic suspensions, they're not autologous cells. And so several primary and secondary endpoints. So these are a couple of the quick surgical videos. We've actually managed to, I would say that managed to deliver these cells with no major adverse events. And so that's the intraop OCT that shows that you actually have a successful delivery of the cells intraoperatively. That's another a video. So some bit of reflux that you see here is inevitable, and this is one of the biggest challenges. And I think we've sort of created an SOP where this is we've minimized this as much as possible. So these are the results. So the autofluorescence is our primary endpoint, and we've shown that, at least in some of our patients, there is a reduction in the area of the autofluorescence, GA area, and then you also show structural changes. So we could see that there are thickening of the outer retina, whether these are cells, whether it is a recovery of the photoreceptors, it's hard to say. So at some point, till we have a histopathological examination, it's not going to be possible to say that. We had a mild worsening of the epiretinal membrane in one eye, but we didn't need to do anything for that. So these are our vision results. So basically, it doesn't matter how much I show an autofluorescence or OCT. If there is no change in vision, it doesn't matter to a patient. So this is our first cohort, which has shown 10, 30, and 11-letter improvement. These patients have now been followed up for about 8 months, following the surgery. The second cohort showed a 15, 17, and a 7-letter improvement about 4 months following surgery. The third cohort has just been done. We don't have, it's too soon to say anything. So in summary, we've successfully completed phase 1 trials, no adverse events noted. What we've noted is a mild preretinal hemorrhage in almost every patient because the retina is very, very thin. This is expected. Mild worsening of ERM in one subject, an average of 14.9 letters of improvement. So we're now going to submit this to CDSCO and get approval for our phase 2 trials. We have some data on, photoreceptors. I'm not going to go into this because that's a long way off. Quickly, the landscape of what else is happening in the world. So Lineage Therapeutics started off in Israel, is now incubated in the US. In fact, this product has been now bought over by Genentech. They had their first 3 cohorts, so they were sort of the leaders in the field, so they had the burden of being the first. So they showed a lot of problems with the surgical delivery. So what they did was to go to the 4th cohort and say that they would do it by an external approach. So this is a lot more complex. You do a pars plana entry, create a partial thickness sclerotomy, pass a band all around the eye until you see it under the macula through a wide angle. And once you see this, you have a foot pedal operated system where you can inject the cells. So they've shown good results with this, but now we're waiting for Genentech to take this forward and tell us what's happening. Luxa is Professor Sally Temple from New York. What they are using is an RPE layer from human cadaveric eyes. And they've just shown their first 9 patient data with really good results. So they've had improvement of about 21 letter improvement on an average. This is the suspension. There is the patch technology, the regenerative patch technology from California. They are using a Parline membrane and implanting this under the retina. And they've had good safety data. We don't know about the efficacy as yet. Dr. Kapil Bharti from National Eye Institute, he is using autologous cells on a patch. And I think they've done 1 or 2 patients and they've had no safety issues. And they are now actually going into a phase 1/2A clinical trial for this. This is as far as the landscape of GA, geographic atrophy. As for IRDs, is there any role in doing stem cells at all? It is there because even if the outer retina is gone, there's a lot of the inner retina that's sustained, and so it makes sense still to do it. So JSIDE is a company that is injecting intravitreally photoreceptor precursors. So this is, it's not mutation specific, does not require immune suppression, and they've actually, shown good results, and they've just launched their pivotal trial. We are yet to see the results of this. Blue Rock Therapeutics, actually this was a big launch in January in the JP Morgan Health Tech Meet. So they are a subsidiary of Bayer who have now started doing photoreceptor work. This is of course going to be a subretinal injection. And I think this is the only ones who are doing subretinal photoreceptors. So I think there's a lot of excitement about what this guy is going to say. So in conclusion, I think this is my personal thing, and that is the treatment for IRDs is not going to be a one-size-fits-all. I think it's going to be a continuum. Early diseases may be, it could be gene therapy. You have multiple mitochondrial boosters like Allegro and so on. Late disease could be cell therapy, optogenetics, Neuralink, and so on. So I do think the future for, it's quite exciting for, these IRDs. Thank you so much. That's where we are based at. There's an incubator in Bangalore called, CCamp, and we are a lab within that. That's my team. And if you're in that part of India, please do come and visit us. Thank you so much.
Thank you very much, Dr. Rajni, for a fascinating talk and very good ray of hope for these IRDs.
Thank you, sir.
Very much. Can we have the...
One injection? For now, 1 injection. So I don't know. Nobody knows whether we need a repeat injection.
All right. Thank you. We will take more questions at the end of the session. Let's invite Dr. Selja for her MACTEL study. Can we have Dr. Selja's talk, please?
Yeah, so thank you again, Dr. Sangwan, Dr. Ken, for this opportunity. And my talk is kind of continuation of what Dr. Ken mentioned in his talk. And I personally am interested in this MAC complex of disorders because unlike the inherited retinal disorders, which have some hope that we could have some treatment in future, once a MAC happens, it stays with the child for life. And it ranges from simple microphthalmos to complete anophthalmos, those with cysts, those with systemic anomalies, and they are emerging as the leading cause of childhood blindness, with the incidence varying between 1 to 15 cases per 10,000 population. Now, if we look at why this MAC happens, there are mainly 2 schools of thought. One is the genetic basis, and other is the environmental factors. If we look at the genetic basis of MAC, there are mainly 4 classes of genes which are involved. One are the transcription factors like the SOX2, the OTX2, the FOXC3, Then there are the signaling pathways, molecules like the TGF-beta and the BMP signaling pathways. The third one, which is very interesting to me, is the retinoic acid pathway with the aldehyde dehydrogenase A13, the STRA6, the retinol acid binding receptors. And then there are some which have unknown function. Now, if you look at the environmental factors, the ones that we can really pinpoint to are the torch infections in the mothers, then maternal exposure to drugs like thalidomide, phenytoin, and carbamazepine during the first trimester. And very interestingly, maternal vitamin A deficiency has also been linked with these MAC disorders. Now, if you look closely here, there is some commonality between the genetic basis and the environmental factors, which links somehow to the vitamin A pathway. And if we see what is the retinoic acid, it is synthesized via multiple step process from the vitamin A. It is a potent morphogen, and it is essential for the embryonic development. It is responsible, in fact, for the development of the normal optic vesicle and the anterior segment of the developing eye. And this is a model just showing the various genes and how they interact in the cytoplasm and the nuclear, and a mouse model where all these genes are expressed in the early embryonic development of the eye. It has also been seen that genetic defects in the retinoic acid pathway lead to very severe kind of MAC disorders. So is there a link between maternal vitamin E deficiency and MAC? And that is what came to our mind. So if we see, there is some indirect evidence like both whole globe abnormalities and maternal vitamin A deficiencies. They are actually more common in rice-dependent communities where there is a chance that there is a likelihood of poor stores of vitamin A. The second commonality is that vitamin A deficiency in mothers as well as MAC disorders both are seen in lower socioeconomic sections of the society. There is some direct evidence as well, both in animals and humans, that mothers who were known to be deficient in vitamin A have given birth to babies with severe MACs. This has happened in piglets. This also happened in some mothers who have undergone bariatric surgery prior to their pregnancy. And some direct evidence of vitamin A supplementation, which has led to a rescue of preventing these changes in both mouse models and some anecdotal human reports. So is this enough? But there are some other direct evidence, like this is a knockout mouse model where retinaldehyde dehydrogenase type 3 was knocked out, and it led to the lethal defect. And maternal retinoic acid treatment in these mouse models actually led to a rescue. Hornby et al., they actually, by seeing the commonality between maternal vitamin A deficiency and consanguinity in a southern Indian population, suggested that there could be a recessively inherited genetic predisposition to the effect of maternal vitamin A deficiency. So here, this kind of like an epigenetic or a environment interaction of maternal vitamin A deficiency in a mother in a fetus who has a genetic defect in the retinoic acid pathway gene. So how did I come to this kind of interest in this part is that it started with the MAC paper of the Bodhi Consortium. And of course, Ken has mentioned that it was the largest study. What we found that around 1/3 of them were severe MAC, and the comorbidities were nystagmus and strabismus, cataract, glaucoma, and retinal detachment. But what was interesting was that we saw some regional differences in our patients. So patients with Sitapur especially, and they had, because that came with a very slow socioeconomic community, they in fact had maximum number of colobomatous microphthalmos, bilateral and anophthalmos. So in our discussion with the group, with my colleagues, with Ketki and Chintan, we had a discussion that Sitapur was also seeing a lot of vitamin A deficiency, and some part of our hospital, a Mohammadi hospital in our shop, we were also seeing vitamin A in cornea as well. So and that's how we kind of explored this association of vitamin A deficiency in MAC, and that led us to the second part of the study, which is again a very interesting finding, that what we wanted to see is it just a coincidence that these two things are coexisting, vitamin A deficiency, corneal disorders as well as MAC disorders. So we conducted a case control study now, and we recruited children who have MAC versus those who did not have MAC amongst the same population in the three institutes, and asked the mothers a questionnaire, a WHO validated questionnaire about vitamin A deficiency in their pregnancy, and we used night blindness as a marker for vitamin A deficiency. Specifically, if you have a local term like Ratondi, which is used in this part of the India, then it becomes the questionnaire becomes much more specific. So we also question ourselves, is night blindness a very good indicator of vitamin A deficiency? Why not do vitamin A levels? But in retrospect, it cannot be possible. It will be very time consuming and money consuming. And the IVACG, which is the International Vitamin A Consultative Group, as well as WHO, they both recommend maternal night blindness as a marker for vitamin A deficiency. They have said that if the maternal vitamin A deficiency is prevalent to the degree of greater than 5% in that population, and if that population, you can go and check for maternal vitamin deficiency, it will not just indicate maternal night blindness, but also correlates with the childhood vitamin A deficiency. It also, the false positive rate in such population is less than 3%, and if you use a local term like Ratandi, then it definitely increases the reliability. So moving ahead, we conducted the study, and as expected, we actually found that the maternal night blindness was around 33% in the MAC group as compared to the 3% in the control group, which was almost 10 times more common in the MAC group. And additional factors which were common, which the multivariate analysis showed, were the birth order and history of spontaneous abortions apart from parental consanguinity. And all of these factors actually are pointing towards the nutrition, poor nutrition in the mothers. Also, we also correlated the severity of MAC with maternal night blindness, and we found that the severe MAC, which was defined as bilateral anophthalmos or bilateral severe microphthalmos, the night blindness was much more common as compared to non-severe MAC. So this led us to the third part, where we are now conducting genetic studies of a 200 patient cohort of MAC patients, and especially looking at the retinoic acid pathway gene to see if the puzzle fits here, that is it really because of the retinoic acid pathway genes that these children are susceptible to maternal vitamin A deficiency, or maternal vitamin A deficiency is kind of working through some other pathways and leading to this kind of defect. So in this study, we would be deep phenotyping all these patients, doing exome sequencing first, and if that is negative, then we'll move on to the whole genome sequencing. And also our US counterpart would be working on the zebrafish MAC models involving the retinoic acid pathway genes. At the end, I want to just conclude, why is this connection between MAC disorders and maternal vitamin A deficiency so important? Because as I mentioned before, the fault in the eye embryogenesis MAC is irreversible. So we have to look at preventive aspects. So the association of vitamin A deficiency and MAC actually provides us a scope for reducing the burden of MAC, apart from, of course, prenatal counseling and testing, and which is also very complicated in MAC because the yield of genetic tests is not very high. We need to know the causative pathogenic genetic variations in our subset of population. There are very few studies from India exploring into the genetic variations in MAC population. We also need to understand the relation between the maternal retinol levels and the MAC and its severity. All this may help us to determine the utility of vitamin A supplementation in populations where vitamin A deficiency is actually frequent. And there are pockets in India where it is frequent, and ultimately it may lead to future public health interventions to prevent MAC as a whole. So my big shout out and thank you for all the participants of the Bodhya Consortium, especially my colleagues, Dr. Chintan from SNC and Dr. Ketki from Sitapur, who have worked with me in this project, Dr. Ken and Dr. Mathur for actually planting the seeds for this consortium, which is the gold mine for all these data, and Dr. Gyan Prakash, who's my mentor in NIH, Dr. Suma Ganesh, who's my mentor in SHOF, and the team Cured, led by Dr. Sangman, and all my colleagues here, especially Ria, my genetic counselor, who is, parallelly running with me in this race. And with that, I would like you all to attend or come to this POSN edition 7th, which will be much more discussion over genetics in a workshop. And that's my time.
Thank you, Selja, for this wonderful talk. And if there is any quick question, we can take. Otherwise, we'll go to the next talk by Abha on corneal replacement. Okay, fine. Is Dr. Benita Kumar here in the audience? No? All right.
No.
Okay, yes, go ahead.
Dr. Shalja, I have a question.
Selja.
Hi.
Yeah,
No. So this was very, very interesting. I think this is learning a lot, really. No, so you mentioned one subject with bariatric surgery.
Yes, there are 3-4 case reports. After
That's very interesting, isn't it?
Mother has severe vitamin A deficiency, giving birth to neural tube defects and microphthalmos and ophthalmos kind of problem.
See, because clearly this is not the low socioeconomic group. It's the other extreme, isn't it?
But after
So,
surgery,
yeah, welfare, yeah.
and they have supplemented in one of the mother vitamin A with low dose, because supplementation of vitamin A during pregnancy is also risky. You can't give it in very high doses because itself it can be
Your name?
teratogenic.
No, but that is something that, so the pediatric surgeons are aware of this now, because honestly, I didn't know this. So then, it's so routinely done now. No, but it's good to keep a lookout on this, especially in the childbearing age for women. Then it's something that they need to be aware also then.
Just vitamin A, they also there are other multiple vitamin deficiencies, zinc deficiency after bariatric surgery.
So again, a neural tube. Okay.
Yeah.
Thank you. That's very
Just one point. Maybe this is also related to the bitoid spots that we see in severe anterior segment dysgenesis. There's something that we showed. There is something to do with retinoic acid pathway in them as well, because you see this foamy kind of a discharge. It's seen in aniridia, it's seen in PTEX2 as well as PAC6. All of these, FOXC1, all of these.
Exactly, yeah. Photoreceptor disorders as well. Precisely, yeah.
I think there is a common factor here, and which is why ABCA4 we don't want to overload them with vitamin.
We don't give them any vitamin, yeah.
Yeah, like the folic acid, you should possibly try and give all the childbearing. Yeah. Okay, interesting.
Compound the problem.
So it increases stress on the RP and can worsen the disease.
What did you forget, Ken?
My computer in my laptop cover.
Did you see it in that room?
No, it's not.
The waiting room?
That's one bag here.
That's mine.
This is your bag.
Okay.
But I'm sure you can't miss your computer cover.
Yes, yeah. Please
Is it
do
there?
find, and thank you very much, Ken, for the contribution. I hope you find your laptop. Yes. Please. Ava, please go ahead.
So after the interesting talk by Dr. Rajni, okay, no problem. His laptop cover has a very good tagline on it, so I'm sure it's not missed. Yeah, the laptop and the cover.
With the cover. All right, so you'll find out. You can.
Okay, so Dr. Rajni Bhattu, your talk gave us a lot of hope for retinal diseases. I would be talking about what we at Shroff are doing for corneal diseases and what in totality today are the innovative solutions for rare corneal diseases. So what are we looking at? We are looking at these kind of conditions presenting with significant surface inflammation, lid abnormalities. We are looking at dystrophies like this where the vision is affected. You really don't want to do a keratoplasty. You are looking at, the patient does have some vision, but you would further like to improve it. We are looking at endothelial dystrophies where, again, now presently the option that we have is only a keratoplasty. So currently the treatment options are you replace the pathological layer, the layer which is pathological, you replace it with donor corneal tissue. But then the diseases that I showed you, are all of them amicable to a corneal transplant? Maybe not. You have multiple recipient factors that you do not, many of your indications may not be suited for a corneal transplant. If you can do a corneal transplant, you still have a very limited availability of corneas. And then you have multiple issues of follow-up, the social economical factors, not forgetting in a corneal transplant, the rate of rejection and failure. So what are the options? I'm going to divide my talk into two things, the stem cell-based options and the bioengineering options. Oh, sorry. These are the options that we are currently looking at, these stem cell-based options, also the biomaterial-based approach, which in further would help correct the diseased cornea or the ocular surface. This started way back in 2000, where May Griffith et al. Worked on the first human cornea equivalence without scaffold. They did manage to do a human corneal trial, but with limited functionality and leading to scaffold integration. The scaffold would not integrate with the cornea. Then came the hydrogel scaffold with better biocompatibility, better viscoelasticity, and better hydration. And also there is electrospinning, where you can create the nanofiber network and further follow it with cells. I mean, you could have corneal stem cell proliferation and migration into these scaffolds as well. So what we are looking at, we have the corneal biomaterial. We are integrating mesenchymal stem cells into them and making it into a collagen-based scaffold with stem cells, hence only replacing the area of the corneal scar. So this is what we are doing. We have the fibrotic cornea, which we have created in the mouse model. We refine the area of the scar, just the area of the scar, and post-excavation, we go ahead, we fill the exosome along with the biomaterial, and then cross-link it with visible light in place, and then see that over time the cornea clears. These are our results. If you see, even clinically on slit lamp, you see the results. On ASOCT, the cornea does come to its original state with good clarity as well as uniformity and surface. Even densitometry is studied where it comes quite close to the normal cornea. You can see we have also compared it with tissue adhesive, which would be the regular mode of treatment. And the tissue adhesive heals with an opacity score of 100 versus the bioengineered cornea, which with an opacity score quite close to that of normal cornea. Similarly, histopathology, if you see the diseased cornea versus the rabbit cornea post-treatment of 90 days, looks quite similar to its untreated counterpart. We also are looking at the full thickness corneal approach, wherein we have the biomaterial that we are studying for a full thickness replacement, that you do not really need the host corneal tissue to be able to support the donor cornea. This is another project that we are now doing with DRDO, and presently it is in the stage of material testing. So this is how we are trying the suturability of the tissue that we have. How suturable, how durable the tissue is, what is its burst pressure, etc., and would it be able to sustain the intraocular pressure. So no full thickness lenticule study has as yet been published, and this is the first of its kind idea that we are working on. Coming to the cell-based approach, for the diseases like I showed initially, we could have mesenchymal stem cells, we have stem cell secretomes or exosomes. They may be derived from the placenta, umbilical cord, bone marrow, or adipose tissue, and what they normally do is they increase cell proliferation, they increase the wound healing, reduce the inflammation and apoptosis of the cells. What is a very good example of stem cell proliferation that has already been used in epithelial regeneration? Further on, moving from Clow to Clet, which was there, and now Slet, small limbal biopsies which proliferate on in situ on the surface of the cornea and allow for reepithelialization of the traumatized ocular surface within, this is a follow-up of a year where you see the patient at initially and at 1 year best corrected visual acuity 612. Also, if you follow it up, you see that the anterior segment OCT of the diseased as well as the healed cornea, the healed cornea is very similar to the native state. Coming to endothelial cell regeneration, this has very nicely been worked on and now has come into, is available for a clinical trial as well, wherein the Kinoshita group has worked on the injection of cultured stem cells with ROCK inhibitor for bullous keratopathy. He has very good results of just injection of cultured corneal endothelial cells, stem cells to reverse pseudophakic bullous keratopathy. Coming to acute SJS, there are significant papers again on the therapeutic effects of mesenchymal stem cells on Stevens-Johnson syndrome. What these cells are doing, like I mentioned in the starting slide, they are preventing inflammation, giving more and more cells the opportunity to come back to the original native state. And there are this is a rabbit study that was done wherein they created a rabbit model of SJS and further worked on in creating the model and then treating them with mesenchymal stem cells, though this is a very small study presently, but then definitely the future has hope for treating acute SJS as well with mesenchymal stem cells. So in conclusion, there is a lot of research and development is needed for refining further corneal constructs and MSC therapies. The key challenges remaining being to address the immune rejection and long-term stability. As spoken previously, there are multiple regulatory hurdles, and definitely we need to navigate the approvals for clinical implementation. With that, I would like to thank my team and open for any questions.
Very good. talk, Abha. Yeah, thank you so much. It's really nice, yeah. So how far are you in the bioengineered cornea now in terms of?
So we're just waiting for approval for the clinical trial. We are making the clinical grade exosomes right now.
Okay.
So we are looking at approval for the clinical trial,
So
human
probably
clinical.
another year or so, we
Okay.
should be doing a human trial.
Yeah.
Brilliant.
And for endothelial injection,
Yeah.
it is now this Orion company
Yeah.
has been
Yeah.
bought by Alcon.
Okay.
So there is likelihood of a global availability of the cell
Endothelial
injection
cell.
therapy
Oh,
for
that's
endothelial
brilliant.
dysfunction. So the regenerative therapies are coming in a good
Yes.
commercial space.
So that's brilliant.
Yes.
So a lot of
So
innovation coming out of India.
your work coming up, hopefully a big company
Yes.
will buy you out.
Thank you.
No, I want to be available for Indian patients. So
Yeah,
thank
of
you,
course,
Abha.
yeah,
Yeah,
thank you,
yeah,
thank you.
yeah.
Please.
Can I invite Ria, please? Dr. Ria Ratna, New Horizons in Genetic Testing. Yeah.
Can we have Ria's slide, the AV, please?
At the offset, thank you everyone and thank you for having me here. I'm going to touch upon the topic New Horizons in Genetic Testing. So genetic testing has been used in several manners as of now. It helps in early diagnosis. So for example, to differentiate between a retinitis pigmentosa, if tested early enough, whether the child will develop hearing loss or not when they come back positive for Usher syndrome. And we know that whole significance because if the hearing loss is tried to be corrected with a cochlear implant early on, it works better than later. It's also being used to prognosticate. So for example, in ARTS-390 has been shown in the Saudi groups and now in Indian groups as well. In the CYP1B1, they have intractable glaucoma that need multiple surgeries. So there's that precise genotype-phenotype correlation that's being used. And in planning treatment for children and adults, for example, RPE65 coming back positive, then there's the Luxturna gene therapy that becomes available. So before we jump into the new horizons, the evolution of genetic testing and what we have known. So a child like such shows up in the clinic and they have a cloudy cornea, but they also have this atypical facies with the downward slanting palpable fissures, mongolite slant, tongue is out, and typical down facies. So we ask for a karyotype which checks for the numerical and structural defects in the chromosome. Now, Down syndrome is very easy to diagnose phenotypically, but why should we do a karyotype then is also because of the possibility of a Robertsonian translocation in which essentially the third extra chromosome is stuck to one of the other chromosomes, and then the recurrence risk for the patient's parents changes significantly, understanding whether it's a free trisomy or not. Then there's first generation sequencing, also known as the gold standard of sequencing, which is Sanger. It is, as of now, only being used for genes that are very small and can be covered all together in one go, or for parental testing. So you have carried out a next generation sequencing test and you want to confirm the presence or absence of the variants in the parents. So a targeted location testing can be done with the help of Sanger. So from there, we move on to next generation sequencing, which we all utilize in our clinics every day, which includes tests like whole exome sequencing and whole genome sequencing, which is now getting into the clinics as well, but has primarily been used in research. And reports of it look like such. We get pointed genetic variations, where exactly in the exon, what kind of mutation, what is the classification of the variant that's found, what is the disease, and what is the, and also sometimes, like in this particular case, you can have incidental findings that can also be reported. Another test that can also be done is polygenic risk score. So this is what it looks like. These are for complex disorders that do not have Mendelian type of inheritance. So you do not have your 25%, 50% risk in the absence or presence of a variant. It's basically a complex of multiple polymorphisms that together give you a risk score. So it can be anywhere from 60%, 90% or lesser that make you likely or predisposed to a disorder. And then there's also multi-omics approaches that are entering the clinic now, which also use transcriptomics and proteomics for deeper insights. So, for example, in retinoblastoma, earlier we only needed to see the RB1 gene to understand whether it is a heritable form of retinoblastoma or not. But now there are markers like TIF1 that have come around that give us more information about how the retinoblastoma is going to behave. But why do we need more techniques than what we already have, and what are these new techniques going to do? So in a setting like ours, like in India, we do not have a centralized healthcare system, and all the costs are borne by the patients themselves. So when we get an NGS negative and we are hell-bound that there is going to be a genetic variation that we have just not caught, we move on to do a chromosomal microarray. All of these costs are all of these tests are high-cost tests and have to be paid out of the pockets of the patients themselves. They lack spatial resolution, and they're a sort of a real-time snapshot of what's happening at that time in the genome of the patient. And there's challenges with variant interpretation. As we know, most of the data that we receive and the classification of these variants that we see is based on the literature from the Western population. So VUS, or the variants of uncertain significance, is seen in our reports more than any other. And also that these techniques are invasive in terms of we need to, in some cases, get the tumor biopsies or a tissue sample to understand deeper about the genetic makeup of that particular disease. In the interest of time, I'm only going to talk about very few genetic techniques that are now making their way and translating from bench to bedside for us. So one of them is liquid biopsy. As the name says, it's a surrogate for going right into a tumor or a tissue sample by using liquid samples. So this could be a blood sample, this could be a plasma sample, it could be CSF samples and such. And it's a non-invasive technique. There is less risk in it and less pain, and it's easy to repeat these. And what you do with this liquid sample, you could isolate what are called circulating tumor cells or circulating cell-free DNA, which is essentially when at the site of origin, for example, if there's a tumor and it lets go of tumor cells and the cells burst, they let out DNA that enters the main bloodstream. So this can be isolated from your genomic DNA and be studied. And there are exosomes that can also be isolated. And this technique has been used actively in in ophthalmology. So in retinoblastoma, Jessie Berry's group in LA has been working on it for over a decade, and she's been taking aqueous humor taps and trying to prognosticate how certain retinoblastomas will be either more aggressive or less aggressive. Our group is trying to standardize this for plasma samples and CSF samples to understand at different time points when a tumor is going to be, how it's going to behave. It has also been used in other malignancies like uveal melanoma. There are biomarkers in glaucoma that are being studied by this technique as well. The other technique that Dr. Neil already spoke about is spatial transcriptomic that's really coming around. So essentially, if you were to imagine a 3D model, suppose, of a tumor, this technique allows the gene expression profile within the tumor sections. So if there were, the hypothesis is, for example, I'll take retinoblastoma, that the tumor originates from the retinal layer of cells and then it proliferates and it starts to get calcified at the end where it started and starts to let off vitreous seeds and it starts to go into the choroid. So some signatures are apparently going to be different in each area. What is making the tumor go either backwards or forwards and making it into an extraocular tumor? And now we have the ability to study all of this. And why this becomes important is because with the help of spatial transcriptomics, you can also get answers like cell of origin, which then becomes important to understand what kind of therapies can come around and what kind of answers you can get based on this information. The other test I would want to talk about is optical genome mapping. This is a non-sequencing imaging techniques that identify structural variants. So essentially, you take very high molecular weight DNA, you enrich it with 6 base pair probes that repeat themselves every 200 base pairs, and they go and attach by a covalent bonding to your heavy molecules, and then you put it into a cartridge for scanning, and the chip essentially linearizes each DNA strand and it makes a picture like such. So it only gives information about structural variants. And why this test becomes important, because when we are deciding testing protocols for patients, for example, you have a child who has a small chromosomal variation, copy number change, for example, a small deletion. Karyotype, even silver banding karyotype has its own limitations, and then chromosomal microarray has its own limitations. Chromosomal microarray will not give you information about the structure of the chromosomes, and if the parents are likely to be carriers of balanced translocations or balanced. So this technique is sort of filling that gap now, where those very micro deletions or duplications that the parents could have in the form of translocations can also be picked up. Yes, and then not to miss, AI in genetic testing is also making its way. How in genetics we expect it to come through is, so all the biobanks and all the repositories that we are now making, the kind of registries we are trying to develop with the help of genotype and phenotype, we will give access to AI and try to develop algorithms that will help in developing machine learning models. And then the first lacunae that I spoke about, that we get a lot of VUSs, we will get better chances of converting these VUSs to either benign or likely pathogenic or pathogenic variants. That process will get much faster, probably without the need for functional studies as well, just with the use of AI training models. And they will help in improving the diagnostic and personalized medicine. Because AI will enter mobile apps as well, one of the problems in India is accessibility to healthcare. So if something can be, if complex disorders can be diagnosed with the help of mobile apps, they can be made available to rural parts of the country. Of course, there are concerns about data privacy and regulatory compliances that need to be kept in mind, but that's to be taken into consideration later. So I want to finish by saying that genetic testing is no longer about diagnosis, it's enabling precision medicine. Multi-omics and AI is getting integrated into the healthcare system, and like Dr. Ken also said, that for all of this to really work out, there should be collaborative efforts of working together with the clinician, geneticists, researchers working hand in hand. So let's embrace this tomorrow. And this is a tiny team that we work with every day, but there's an army of scientists and clinicians at Shroffs always supporting us. So thank you so much.
Thank you very much, Ria. We have time for a few questions or comments if anybody wants to, for her talk or for any other talk. In the meantime, may I request all the speakers to come up and take a picture? Those of us who are left. I know Ken is shaken by his laptop. All right, if no questions, please come for a picture quickly, and then we can vacate the hall.